Teaching professor. He is the founding director of the Multiple System Atrophy Clinic here at UT Southwestern. Welcome, Dr. Vernina.
Thanks, everyone, and thanks for coming. Again, we like to have these sessions fairly frequently, but the topics are entirely up to you. So if there's other things you want to hear about, please let Chidima or Amy know, and we can add it to our schedule. As I mentioned earlier, maybe before some people got on, the MSA Coalition is having their virtual conference later this year, the 2022 virtual conference, September 29th to October 1st, so you can mark your calendars. And the UT Southwestern team, all of us here are going to help them organize that. So you may see more of us later in the year as well. And I think they really want to focus on the multidisciplinary care that is really what we're trying to accomplish in many places around the country. So welcome to those of you who are part of our UT Southwestern Clinic, who've been in research studies with us, and all visitors or anyone else who's joined to spend the 45 minutes or so with us. We have to jump off promptly because we have MSA Clinic patients to see this afternoon. All right, so today's topic is an update on research. So I like to give an update at least once a year on what's happening. And it's fortunate that we have things to talk about. For many, many years, we recognized that MSA was a bad disease and was progressive, and we were starting to learn how it affects patients and what the cause is. But we're in a new era now in the last few years, really, where actually clinical trials that not only look at medications and other options to treat symptoms, but also those that are targeting the actual disease. So we'll talk about some of that today. So research in MSA is got a lot of different aspects to it. There's been a lot of research on the cause of MSA, and I'll talk about that in a moment. There's more and more research on biomarkers. Why is that important? Biomarkers are either lab tests or imaging or other types of laboratory tests that can be done to help confirm the diagnosis of multiple system atrophy. Those of you who have experience with the disease know that there is no definitive test besides looking at the brain after death to be entirely 100% certain about the diagnosis of MSA. But that is changing, as we'll talk about in a little bit. There are preclinical studies going on. So anytime a new treatment comes along, it needs to be studied before it's given to patients. And that could include looking at these drugs or approaches in cells in the dish in the laboratory or in animal models. So in mice, for example, who have a animal model that replicates some of the symptoms of MSA. And then there are clinical trials, which could include drugs that treat symptoms or drugs that are supposed to slow down the disease. And we're also doing research, as you know, in our setting on how we can best deliver care to patients with MSA. And thank you for those of you who are part of that. Also want to give a little background as we talk about clinical research and clinical trials, that you may hear these different terms about the phase of the study. So I just want to touch on that real quickly for your education. So as I mentioned, preclinical studies are those that where you're studying a treatment, but not in people, but rather in animals or in cells or in the laboratory. And that's required to make sure these drugs do what you think they do and that they are safe. And then there's something called a phase 1 study. So anytime a new drug is developed, the FDA requires a phase 1 study. This is for safety reasons. And the participants in phase 1 studies are normal, healthy controls. So there are volunteers, we're grateful for them, who volunteer to take a brand new drug and report if they have any side effects, just to make sure that the drug at the doses that we might use in patients is safe. So after phase 1 studies are done, you move the drug will, assuming it's found to be safe, and it was moved on to phase 2 trials. So phase 2 trials are what's sometimes called the pilot trial or the preliminary trial. And that's where the patients with the actual disease under study get the drug for the first time. Oftentimes in a phase 2 study, there may be different doses. So some patients may get a low dose, some patients may get a high dose. Again, looking for safety primarily, looking for side effects, but also still looking for effectiveness. So does the drug seem to do what we think it does? But these are usually smaller studies because they were looking for safety and looking to see if a larger, more expensive study is going to be feasible. And if that goes well, then the drug may move into phase 3. And this is what we really call the definitive trial. So these are double-blind, placebo-controlled trials with enough patients to really show a benefit of the drug. And the FDA requires a phase 3 trial showing benefit. Sometimes they require 2 phase 3 trials showing benefit before they'll approve a drug. Fortunately, MSA is considered a rare disease, and so the FDA has special rules for rare disease drug development so that the drug could potentially get approved after a really good phase 2 study or after a single phase 3 study, unlike other drugs like for hypertension or diabetes that require big studies at least 2 different phase 3 studies. Okay, so that's the background. Let me give a bit more background about what we know about what causes multiple system atrophy. So we now know that this particular protein called alpha-synuclein is part of the problem. What's really interesting is that alpha-synuclein is a normal protein made by nerve cells, by neurons in the brain and in other places. So, but in the case of multiple system atrophy, alpha-synuclein builds up in the glial cells. The glial cells are the supporting cells of the brain and spinal cord, and they're not nerve cells, and they don't make alpha-synuclein. So how does the alpha-synuclein get in there? And that's been an area of lots of research. But suffice to say, the alpha-synuclein protein has to get out of the nerve cell and pass over to the supporting cells. And why does that happen? And how, we don't really know. But if we can intervene and prevent this protein from getting into these supporting cells, we might be able to stop or reverse the multiple system atrophy. In other diseases like Parkinson's disease, the alpha-synuclein protein stays in the nerve cells and builds up there and causes disease. That's why MSA is different than Parkinson's disease pathologically. But eventually, if those supporting cells get sick and die, then the nerve cells themselves start to degenerate. And in MSA, this typically starts in certain brain regions, like the brain stem, where a lot of autonomic control centers live, and the cerebellum, the balance center of the brain. So that's why balance and autonomic problems are very common in multiple system atrophy. And then finally, we think there's a role in inflammation. So alpha-synuclein is kind of toxic, and so the body's immune system will start to get excited, and there'll be inflammation around the areas where alpha-synuclein is accumulated, and that makes the nerve cells unhappy. Sometimes we call that oxidative stress, and you can see in the cloud there in the middle, all these things that are causing stress to the nerve cells while this alpha-synuclein builds up into these blobs that are hard to dissolve, and they kind of stick there. Okay, so that's the background. In terms of biomarkers, we've depended a lot on our clinical judgment. We talk about symptoms, we do an examination, but we've known for a while that brain scans, the MRI scan, can show some characteristic features in multiple system atrophy. We use that as a biomarker, either changes in the brainstem, like is shown in this picture, but we've more recently seen some other typical changes in multiple system atrophy in various other parts of the brain that help to distinguish this from Parkinson's disease or other similar disorders. So we have the MRI scan, and that would be obviously very important if we can have a suspicion and then some tool to make the diagnosis sooner, so that we could start an effective treatment sooner than later, before the disease is full blown. We also now have the ability to look for this abnormal protein, alpha-synuclein, in the skin, in the blood, and even in the spinal fluid, as I'll show you in just a minute. I think my slides got a little bit out of order. Oh, goodness, hang on a second. I'm going to turn my slides off for just a second because I seem to be missing something here. Second. Talk among yourselves. There we go. Where that went away. All right. Too many things to put here.
Hi, this is Amy. I'm going to put my email address in the chat. So if anyone has any questions or would like to be added to our monthly list for invitations, just let me know.
Okay, so I'm back. Can you see the slide now? Or am I on the right side? Can you not see it yet?
Right, it's not showing yet, the share.
Gotta go back to Zoom. So many buttons here. Okay.
I know, too many.
And we should be here.
Okay,
Now
yeah,
what are
we're,
you seeing?
yeah, just go in presentation mode.
Switching over. There we go. Okay, so in the I just want to mention these couple of really exciting new things that are happening, and this is research, although not treatments, but really biomarkers. So there are now ways to detect this protein alpha-synuclein in the skin, and so without having to biopsy the brain or the spinal cord, we could potentially take a small piece of skin, much like the dermatologists do when they're biopsying, something funny on the skin, small little piece of skin, send it off to the laboratory where they can look at the little nerve fibers that are present in the skin. And you can look at the number of nerve fibers, which was a test we've done on skin biopsies for a long time. But now we can stain using a microscopy and look for alpha-synuclein in those little nerve fibers. That's kind of shown here a little bit.
Dr. Bernino, I'm sorry, but it's not in presentation mode and the slide is just a little smaller.
How about now?
Hold on.
It's hard to tell which.
You
seeing it?
I see it, but it's like we see your next slide and then it's your slide and then all these little buttons at the bottom.
Oh, still like that?
Yeah.
All right, I don't know what's going on.
Okay, yeah, I think if everyone is okay and they can see it, then I'm cool with it. Yeah, I think it's, you had it in presentation slide mode, the first one, but.
Yeah, let's go start it again.
Okay.
All right, start. Start my slideshow. Okay,
Okay, yeah, we're good.
Okay.
Yeah, this is good. Okay.
Okay, back to the slide. So what this is basically showing, you don't have to see it in detail, but that you can see the little nerve fibers here in green and the alpha-synuclein protein in red, and see that this alpha-synuclein is inside the nerve cells, and that's a characteristic feature not just of MSA, but of any of the alpha-synuclein disorders, so multiple system atrophy, Parkinson's, and so on. So that's a good way, and we've used this test already to help to confirm our diagnosis. But now they're looking, there's a study going on, and some of you may have heard from Amy about it, that there may be ways to take this information and separate out between MSA and the other alpha-synuclein disorders like Parkinson's disease based on what's going on in the skin biopsy. So that study is going on, and we're looking for some MSA patients who would be willing to have a skin biopsy for research purposes. The other studies that are that have been shown up just in the last year and a half to 2 years is some ways to identify multiple system atrophy in the through spinal fluid tests. So shown here are 2 things that you can measure in the spinal fluid, something called NFL. That's not the National Football League, but it's neurofilament light chain, which is a protein, again, normal brain protein. But you can see that in MSA patients, the amount of NFL in the spinal fluid is much higher than we see in control patients, Parkinson's patients, Lewy body disease, or even pure autonomic failure. So that's one way to differentiate, although neurofilament light chain protein is elevated in other things like MS and stroke and so on. But you combine that with another test. This is what's called an alpha-synuclein seeding assay, which allows you to look at alpha-synuclein in the spinal fluid and see, number one, if it's there, and all of these, if it's there, and then whether it aggregates or clumps up when you look at it in the laboratory. And what's interesting is that although MSA patients have a lot of alpha-synuclein in their spinal fluid, it doesn't clump up. This is a clumping up assay like it does for these other disorders. And you can see that these separate pretty well. So between these two tests, or three tests, if you will, we believe that these tests will become part of our standard practice in making a more definitive diagnosis of MSA. Although as experts in multiple system atrophy, as you know, we oftentimes we're pretty sure we know what we're seeing. Okay, so let's move on to therapeutic trials. So there are still some studies going on. There will be some more studies on drugs to help treat orthostatic hypotension. We're not going to talk about that too much today, but we're really interested in these in drugs that we call therapeutic, or that they would potentially change the course of the disease, slow it down, stop it. I guess we could hope reverse it perhaps at some point. And so there's, as you can see from this list, and I got this from the MSA Coalition website. They keep it updated pretty well. These are all the various drugs and companies that are working on this. There are also, these are other studies, and you can see studies on skin biopsy tests and so on. I'm going to skip over that. So what about the clinical trials? So let me give you some updates on things that have been going on. So at the Mayo Clinic up in Rochester, as many know, they completed a phase 2 trial of stem cell treatment. So what are stem cells? Stem cells are taken from a person's body. These are autologous stem cells, are taken from yourself, and then they take those cells, usually from a biopsy of the abdomen, taking some fat out of the belly. They take it to the laboratory and they select out the small little number of stem cells. Stem cells are cells in the body that could, they haven't really decided what they want to be yet, so they could turn into anything, including nerve cells. So they take those cells in the laboratory and they treat them with certain chemicals and growth factors to make them kind of turn into nerve-type cells. And then they inject those neuronal stem cells back into the spinal fluid, basically through a spinal tap. And when they did the phase 2 study, they showed that there was some, again, phase 2 study is not a lot of patients, really looking more for safety. They found that it was pretty safe. There were some side effects, and it seemed that the progression of multiple system atrophy slowed down some in the patients who got the stem cells. But ultimately, there wasn't a change in survival, meaning those patients didn't really necessarily live longer. So now there's a new study that's just gotten underway, which is, I think, still considered a phase 2 study because they're still making some adjustments to the way that they develop the stem cells and how many of them they give. But there's very strict criteria to get into that study, and I know I've talked to some of our patients about it. Okay, what about the Biohaven studies? There was a study that we were part of here at Southwestern using a drug called verdiperstat, which is a drug called a myeloperoxidase inhibitor that's supposed to reduce some of that stress, that oxidative stress that your nerve cells are under when you have MSA. And so the big phase 3 study went along. We recruited quite well. And we were very sad to hear last November that this study showed that there was no benefit. The patients who got the drug compared to the patients who got the placebo were really not any different in terms of how fast their MSA progressed. So that was quite disappointing. But these things happen, and there's already been other drug studies for MSA that didn't show a benefit. Research is important. We wouldn't want to use this drug unless we really saw that it had a significant benefit. There was also a symptomatic trial of a drug called ampreloxetine. And this was a drug not for treating the underlying cause of MSA, but treating the orthostatic hypotension in MSA. And unfortunately, this drug also showed no benefit in terms of treating the NOH symptoms. So that was disappointing. But we just got a news release earlier this week from the company saying that as they've analyzed this data and looked at those patients who had multiple system atrophy compared to patients who had other types of orthostatic hypotension, it seemed that the MSA patients actually did seem to get a benefit. That's still a work in progress, so we don't know the final answer yet. But the drug is, the company hasn't given up on this, and we actually, in our experience, think that it would be a helpful drug to treat the orthostatic hypotension. So a symptomatic drug that's still being developed, and there are others in that coming out as well. Okay, so what about starting soon? That's what we're really interested in. What's next? We've maybe talked before and maybe even last year about what other drugs might you use to treat MSA and try to slow this disease down. And one of the ones that I'm very excited about personally is are drugs that are actually monoclonal antibodies that target this alpha-synuclein protein. Remember, that's the protein that nerve cells make, but then it gets out and builds up in the supporting cells in the brain. So if we could send an antibody in there that would grab that alpha-synuclein while it's outside the cells and prevent it from moving across and getting into the supporting cells, that might stop the progression of the disease in its tracks. And indeed, in animal studies, again, preclinical studies, it was shown that this type of treatment really stops the alpha-synuclein from spreading, and also even kind of sucks it out of the cells where it's already gotten into. So that was very exciting. So multiple companies are working on alpha-synuclein, and many of the drugs have passed the phase 1 phase. So they've been given to control patients and they appear to be safe. So now phase 2 trials are coming. The other approach that I'll talk about also is drugs that modulate iron in the brain. So iron, of course, is a normal mineral that we all need, but it appears that the process by which the alpha-synuclein protein gets clumped up or aggregates requires it to bind to iron. And so if one could reduce the amount of iron specifically in the areas of the brain where alpha-synuclein is causing mischief, that might do a lot to help slow down the disease or perhaps even reverse it. We also know that iron can participate in that oxidative stress pathway. So reducing iron levels in the brain is something to think about. And again, phase 1 studies have been done with this drug, and it seems to be helpful. And it's not a drug that's so strong that it reduces the body's stores of iron, because you wouldn't want to get iron deficient, but it's strong enough to get iron out of the areas where it doesn't belong. Okay, so here's a little bit more about that. So how does monoclonal antibody treatment, how would that work? Monoclonal antibodies are kind of blood proteins, so you really can't give them as an oral pill. These have to be given by infusions. And there's lots of examples of monoclonal antibody treatments that are being used for rheumatologic diseases like rheumatoid arthritis. And I'm sure lots of people see the advertisements on TV for Humira and other things for diseases like that. Those are monoclonal antibody treatments. So this is not a new type. But monoclonal antibodies against alpha-synuclein is a new development. So these drugs have been developed for over the last couple of years. And in most cases, the treatment would need to be given by an intravenous infusion about every 4 weeks. Again, there's a number of companies that are working on this, but that's a typical treatment pattern. Most of the studies are going to be 1 year in duration. Based on past studies, it seems that you have to do a study for about a year before you can tell whether MSA is progressing or not. Because although it does progress rapidly, it takes a year to really differentiate whether someone's stable or progressing. So these studies will be 1 year in duration. Double-blind studies, meaning that there'll be randomization. Some people will get the real drug, some people will get a placebo or saline infusion type of thing. Okay, so remember I talked earlier about the levels of alpha-synuclein in the spinal fluid. And so these studies, all the ones I've looked at, typically include brain MRI scans at the beginning, at the end of the study to see what the brain looks like. But most of them also include spinal taps. So that may not sound very appealing, but they're in there for 2 reasons. Number one, so that we can use these new biomarkers to make sure that the patients that are getting treated are actually MSA patients and they actually have alpha-synuclein floating around that's causing problems. We hope that if this sort of treatment is effective, that the follow-up spinal tap will show there's a lot less alpha-synuclein that's in the spinal fluid as another way to indicate that the drug is working. So again, there's been some phase 1 studies on monoclonal antibodies. Phase 2 studies are coming very, very soon. So 2 companies are involved in this space. I can say one of them is Lundbeck because they made a public announcement about their study. We are not participating in that one, but we are participating in a very similar antibody study with a different company that I, for confidentiality reasons, can't name at the moment, but the word will be out very, very shortly. And we will be participating as one of the early sites in a monoclonal antibody study, which is exactly this kind of thing, infusions with some spinal taps to be, one at the beginning, one at the end of the study a year later to monitor the progress. Most of these studies are looking for patients who are relatively early in the course of MSA, and that usually means still walking, but you can use some assistance, but walking independently, by themselves or with a cane or a walker. So all the more reason to stick with your physical therapy and keep walking. And then some of the studies have other, obviously there's lots of inclusion and exclusion criteria, but that's the main one that we look at. What about this iron drug? So Alterity is the company that's developing this drug. And again, I'm giving you public information here. Their drug is called ATH434. This is a drug that binds up the iron and pulls it away from the alpha-synuclein protein so that it can't bunch up and cause stress on the nerve cells. So again, this is a phase 2 study, but this is an oral drug taken twice a day. So for those patients who can't tolerate intravenous infusions or live too far away to come once a month, this is another study that we hope to participate in. Again, a 1-year study, randomized. My understanding is that this one will be randomized in what they call a 2-to-1 ratio, meaning that twice as many people will get the drug as the placebo. So that's kind of nice, but the patients and the investigators still won't know which drug people are getting. It appears that, and again, we don't have the final protocol yet, that this will be limited to MSA patients who have the Parkinson's type of MSA, the type P MSA. And they're going to be measuring iron by using a brain MRI scan. This study won't involve spinal taps. So again, phase 1 studies have been done, animal studies have been done to show that this is a treatment that's worth pursuing. And of course, so were the other drugs that were tried in MSA and ended up not showing a benefit. So that is the nature of research. But we are, as part of our commitment to our patients, trying to be involved as early as we can with the latest research therapies that are available. And should go without saying that we try to give this information out so people can have their options as far as participating in research or not. But ultimately, that decision's up to you if you were to meet the inclusion criteria about whether the study is right for you or not. Okay, so that's the update. Hopefully that was useful for people. I have our research coordinators here as well, but I'll be happy to take some questions from the audience or from anybody else that's out there. Were there some chat questions, Chidima?
Right now, no. What did you want Steve to just kind of introduce himself and
Sure.
his role in the okay, Steve, the floor is
Sorry,
yours.
I'm eating chocolate right now. Sorry.
Share, please.
You caught me right when I was eating chocolate. Hi, I'm excuse me. I see a lot of people on here I don't really know. I'm Steve Hopkins. I am Dr. Vernino's clinical research coordinator. I'm the manager of the neuromuscular research going on. The 2 trials that Dr. Vernino was saying that we're going to potentially come here, I've been working on one of them pretty quickly, the one he couldn't really talk about too much, but so hopefully that one will be moving along pretty quick. The other one, it's in the early phases, but Amy, I also work closely with. So if you want to get in touch with me or Amy in regards to where they're at, give us some time. But hopefully in the next few months, you'll start hearing from us about them being closer with an approval date on when we can start getting patients into to see how they're doing, to get them into trials potentially.
Okay, thank you. And
So
does
there's a-
anyone, go ahead.
Yeah, there's a chat question here I'll try to address. So there was a question about my opinion on stem cells that are intranasal administered. And then the question about what does mesenchymal mean? So there's different kinds of stem cells. Mesenchymal stem cells refers to the stem cells that are derived or that are collected from connective tissue in the body. So, and in the case of the Mayo study, they're using mesenchymal stem cells that are taken from the abdominal fat, because most everybody has some extra abdominal fat that they can donate for that purpose. So that's just the type of stem cell. As far as intranasal versus intravenous versus spinal fluid administered stem cells, I'm not an expert in this area, but it's been shown that the stem cells that are given in the blood, like intravenously, only a very small portion of those can make their way into the central nervous system, the brain, spinal cord. Those that go intranasally, the concept is that at the top of your nose, there's a thin, there are a lot of nerves that come through a thin area of bone, and those olfactory nerves go directly into the brain. So the idea is that if you can get those stem cells kind of up in that area, that they might have a way to better find their way into the central nervous system. I don't know that that's been very well proven, but it's attractive to think about intranasal administration because it's so easy. Now, let me make just a general comment about stem cell therapy, because I think it's an area where patients who are looking for answers, dare I say desperate, may fall prey to bad medical practice. So there are stem cell companies that pop up all over the place. Some of them are offshore, Bahamas, Cuba, and so on, and they offer stem cell therapy for neurodegenerative diseases. There are some that even the United States that say we offer stem cell treatments, and they give them intranasally or intravenously and so on. But those are not really what we're talking about, because you can collect stem cells from anywhere in the body, give them back to the patient, inject them into the skin, shoot them up your nose. But if those stem cells haven't been properly conditioned, trained to produce the right kind of chemicals to play the correct role, they won't be helpful. So in the case of the stem cells in the Mayo Clinic study, they actually found that the stem cells that seemed to be most effective were the ones that were producing certain brain growth factors, and those growth factors seem to make nerve cells happy. So it's not that the stem cells go and take the place of the nerve cells that have died. They generally are secreting various nerve growth factors that the nerves like, and it makes the nerves happier, and they live longer. So one approach that's being taken now is whether we can replace stem cells with actually just produce those nerve growth factors that are needed and give those to people directly. Just a cautionary tale about stem cell therapies. I've had patients who've even gone to China and sometimes come back very ill because of whatever the treatment is they got. We don't have effective treatments for MSA right now, so I won't judge people or tell them what they should or shouldn't do, but use caution if you're working outside of a really well-designed clinical trial. So Joy asked whether skin biopsy studies or spinal fluid testing can give us a conclusive diagnosis that eventually won't require a brain donation, or more importantly, could help you make a definitive diagnosis while you're still alive. That's the goal. It looks to me like that answer will be yes, but again, this is still in the research phase. I'm optimistic that one or both of those done, gathering that data appears to be pretty conclusive. There'll always be patients who kind of fall in the middle, but that's one reason we're doing the skin biopsy study to gather more data on this. And there are still spinal fluid studies going on, but really the issue of the spinal fluid testing is that they're at the point of just trying to commercialize that so that that test is available more readily to everybody. It requires a spinal tap, but that is certainly something that would be worthwhile if you could make an earlier and definitive diagnosis, and if there are treatments available.
Okay, does anyone have any other comments, questions, or concerns?
I see that Carol joined. I just want to let her know that I did put in a plug for the fall conference.
Okay, I see one from Richard Gordon. Is this study in Utah available to people out of state?
You'll have to give me a little more information about what Utah study you're talking about.
Give us a little more
Here,
meat,
just
Richard.
type it in. Okay,
Type what?
any study, just put any study.
Oh, you're in Utah, and you're asking if studies are
No,
available.
no,
Oh,
in New Jersey, just put, I live in New Jersey.
oh. Oh, are any studies available to people out of state? Well, potentially yes. So a lot of times when we talk about a research study, the first step is inclusion and exclusion criteria. Are you, do you meet the criteria for the treatment? And then we have to have a very frank discussion about what's involved. How many studies are involved, what tests are involved, because there has to be, commitment by the patient that you're going to participate. So, if you live out of state and the study requires you to have a visit at the study site once a month, or once a week, that's probably too much traveling. If it's a study visit every 3 months, maybe that's doable. Now, I will say that most of these studies I talked about today are multi-center studies. The ones on alpha-synuclein antibodies and the one on the iron drug. They aren't started yet, but those and many other of the research studies are multi-center, meaning that even if you're not close to Dallas, there might very well be a research site near you, at, an academic center in Boston or New York or what have you. And one good resource for that beyond the MSA Coalition would be a website called clinicaltrials.gov. And you can go on clinicaltrials.gov, type in multiple system atrophy and see what's there. That site will list not only therapeutic trials like what we talked about, but also biomarker studies, natural history studies, and so on. So, and certainly if you have questions about those, talk to your doctors about, what might be appropriate for you. But the long and the short of it is, if you live out of state,
Pennsylvania.
what's that? Oh, okay. But the long and short of it is, if you live far away, then you have to really look at what's involved in the study very carefully. How many visits are there? What travel is needed? Whether there's any assistance in the study for travel and so on. So someone here mentioned they're going to be part of the stem cell study. That's good. So you're interested in the biomarker studies. So I know that the skin biopsy study that we're part of, the main study site is actually in Boston. So if you're up in that
Are there any? Ask him if there's any in Philadelphia.
I don't know off the top of my head. We again, clinicaltrials.gov might help. And Amy and I can look into the study sites for that study and see if something in Philadelphia. I don't know off the top of my head, but have to do a little investigation.
So can I add one thing? So I'm Carol Langer. I'm with the
Yeah, of
MSA
course.
Coalition. Thank you for the plug for the conference. We really appreciate your being part of that for us or with us, and also for the plug. A couple of things about clinicaltrials.gov. We have actually found what I would call a stem cell trial on clinicaltrials.gov that I personally thought was not really legit. And so one of the things that you need to look at, particularly for a stem cell trial, and we get questions about this all the time, is look at who the principal investigator is and look at that individual's qualifications. In the case of the one that was on clinicaltrials.gov, the PI was I believe either a dermatologist or an ophthalmologist. Not the best choice for somebody who's treating your neurological problem. So there are some red flags that you as patients or care partners can look for there. And, you know, I completely understand how people want help and want to cure their disease, but you don't want to make it worse.
Yeah, I think good point, Carol. So clinicaltrials.gov is just a clearinghouse for
Mhm.
anything. And it doesn't, you know, they're not trying to, the government is not trying to endorse or prove or state that these
Or vet. They don't really
they don't
vet.
vet them. Yes,
Yeah,
thank you.
yeah.
That's the word I was
Yeah.
looking for.
I do that.
They're not vetting them. They're not saying it's good research. They're just saying there is a trial going on. That is all it says. And you really have to, you know, gather the information and be a good consumer of that information so you know what your options are. And again, if you have a good relationship with your treating physician, be open about it. Say, I saw this study and I'd like to do it. What do you think? And you know, they can help you kind of decide whether it makes sense or not.
Mhm.
Hi, Dr. Vernino. It's Karen and Dale.
Hello.
Are you going to be contacting your previous MSA patients that you had regarding the upcoming studies?
Yeah, we sure will. So one of the advantages of having a formal MSA clinic, and you know, with the help of the MSA Coalition, we're trying to encourage more places to do this, is that we have a database or a record of all of our patients and kind of where they're at. So as soon as we get the inclusion criteria, we can quickly look through the whole list of patients and see, you know, who's likely to be eligible and start reaching out to them.
You already signed up at University of Pennsylvania.
Yeah, exactly. So many of the academic centers will have a database. So as soon as a new trial comes out, they'll easily be able to review the patients that they already know about and see which ones might fit the bill. So yes, that's an important part of what we do. And I will say also that if you happen to have participated in a prior study, that's fine. I believe when I look these things over, you know, they obviously required you can't be in two clinical therapeutic trials at the same time, and they typically require, you know, 1 to 3 months of washing out any previous investigational drug before you can start something new. But given that all the studies we were involved with have now closed up, that shouldn't be a problem.
It looks like Joy Steen posted some good information. She said that her husband participated in a clinical trial. We use a profit called Angel Flight. So please take note. Angel Flight to fly us to our trial site. This means that we didn't have to fly commercial during the height of COVID. Just sharing this information in case it can help anyone. They fly medical patients who can't fly commercial due to health reasons. Thank you, Joy Steen, for that information. And Amy, I'm gonna let you take it away and round this up. Thank you, Dr. Vernino, for your presentation. Go ahead, Amy.
I just posted to the chat that if anyone would like a copy of these slides or the recording from today's session, if you will email me, I'd be happy to provide that for you. And my email address is in the chat. And we should be having our next educational session on May 17th. This will be about sleep and breathing disorders in patients who have MSA. And I will be sending out invitations for that towards the end of this month. If you would like to be a participant in that, please send me your email information. Those that are UT Southwestern patients, I already have your contact information. But anyone from the outside, if you'd like to be invited, just send me your contact information. Thank you, Dr. Vernino. Thanks everybody for participating.
Thank you.
What's her email?
It's in the chat, amy.conger@utsouthwestern.edu.
Our emails here are pretty easy.
Yeah.
First and last name, utsouthwestern.edu.
Thank you, everyone, for joining us.
Thank you.
Good to see everyone. Thanks for joining.
Have a good afternoon.
Bye.
Have her email?
Yeah, it's in here. It's in the chat.
So let me write it down. I'll write it down if you
There it
get it.
is. Amy Conger.
AMY.
All right, Shaneem, I'm gonna leave the meeting running for a little while so that people can get that information out of the chat.
Okay.